we would like to introduce and welcome Dr. Björn Rotter, co-founder and Chief Technology Officer for GenXPro. It's a startup company located in Frankfurt, Main, and it's operational since 2005 in this area and doing great work in the field of precision medicine as well as precision agriculture. Let me introduce Dr. Björn Rotter shortly. Dr. Rotter completed his BSc from College of New Jersey. Afterwards, he was enrolled for his MSc thesis at the J.W. Goethe University in Frankfurt. After completing, he went to Paris for his PhD thesis, where he did his PhD from 2004 till 2004. And immediately after completed his PhD work, he co-founded GenXPro together with his colleagues. And since then, he is in Frankfurt region. And so we are very happy. Welcome, Dr. Rotter. Maybe to begin with, I will like to ask you, how do you think that precision agriculture and precision medicinal technologies are important in the context of pandemic as well as a developing country like India?
Mm-hmm. Well, maybe what we see now in the recent years, we see that there's the gap between the scientific know-how in India and the rest of the world is I think very, very close now. Mm-hmm. And we see a lot of very, very skilled people in India, both in the labs, but also what is also very important in this field that we are active in is the bioinformatics field. And mm-hmm. both of these areas, India has a very strong background already. And then, of course, so this opens up the opportunity to work in this field at the forefront, I see. Mm-hmm. And then, of course, India has also an increasing know-how in the field of pharmaceutical trials, etc. Also, this is important in order to get products and diagnostics going into use to be used finally in the clinic. Mm-hmm. And for the plants, India is an enormous country. And if we look at the potential of medicinal plants and also, of course, regular crops, I think also India has a rich source of biodiversity, which is also an interesting field where a lot of treasures can be found that if they are explored correctly.
Mm-hmm. Thank you. So when you say that India has great biodiversity, there is one particular medicinal science, Ayurveda, which is very ancient and very dear to Indian subcontinent. And we'd like to know what prospects and what kind of opportunities you see for selective plant breeding technology for the field of Ayurveda, where there are certain aspects which are very important. Let me explain a little bit. In certain areas, certain Ayurvedic plants are grown, and at a particular season, certain chemicals are present in that plant. These plants are harvested at that particular time point to have that medicinal value. Also, the area or the soil in which the plant is grown is also important. Since India is a huge country, we have Himalayas where the temperature is temperate or colder versus Kerala where it's most humid and warm. And that also defines the differences between the medicinal value of a Ayurveda plant. So how do you think your breeding technology could be good in this particular context?
Well, maybe I spend some time first on explaining what we are doing. So we are experts for so-called next-generation sequencing technology. So we are using specific technologies that are very cost-efficient and have revolutionized the whole life science area in the last 10 years to sequence basically the DNA of any organism that where DNA is available, And also what we are experts in is also to look at the gene activity. And we are using both these levels of information and combine them in order to find genes of interest and to identify genes that are, for example, responsible for a specific trait that is important. For example, these traits could be a resistance towards a pest or a better resistance towards drought, stress, etc. And so we use this technology, and of course, we see that we could use it, of course, as well in all kinds of breeding applications, and also for the better understanding what is happening in these medicinal plants. What are the compounds that make the value of these plants? What are the active ingredients? And so I also see a big potential in better understanding the biology of these plants, and also what are the secrets behind these findings that are very well known since a very long time already.
In the recent context, food sustainability has become the big question, or the certain questions for world, especially for in the Indian context. When I see that to manage such a huge population, food sustainability will really give a boost. And how do you think that selective breeding could help in bringing this particular aspect in Indian context?
Mm-hmm. Well, selective breeding, maybe I can just explain a bit what is selective breeding in comparison to genetically modified plants. So selective breeding is basically an acceleration of the traditional breeding. Traditionally, the breeder is looking for a very interesting trait, let's say again the resistance towards a certain stress, and then he is using this plant that has this trait and crossing it with his already established plant lines. And then he has to look which of the offspring of the plants has the trait, but also does not lose too much of the original line that he was breeding for before many years. So because usually these plants that have the trait are maybe very tiny, do not have a lot of yield, etc. So you always have to. This is what takes so much time, because then you have to test for all the different traits that you have bred before, maybe hundreds of years before, if they are still there or lost. And this takes a lot of time, because you have to see, is there still the resistance towards the stress that you knew before? Is there still the resistance towards the fungus that the original plant had before? And is in addition now this trait that you are interested in integrated in the genome, and only this trait and not too much of the other traits, because they are maybe detrimental for your yield. So this is the big problem since thousands of years of breeding. And what we can do is we can find the genes that are behind the trait of interest in a faster way. And by doing this, of course, this opens up a lot of opportunities for selective breeding that will not use genetically modified plants. So the fast way to do this, of course, is if once you know the gene, you could cut it out of the plant, and maybe even from another plant that is not related to the crop that you're working with, and introduce it into your genome. That would be of course, the genetically modified plant. But this is not what everybody likes, and so this traditional way that I just described uses the natural resources that are already available in the plants and introduces them, and it does it in a faster way. And the acceleration comes because we can identify at an early time point. We don't have to do all the testing of the crop under the different conditions, but we can very early on look in a very small leaflet already if the gene that we are interested in is available in our offspring. And we can then do the selection based on these molecular markers, this molecular information that we have. And that's the big difference. And yes, I think there is a big potential for all breeding applications, and of course including India. We ourselves have also worked with chickpea, with a research institute in Hyderabad already, to improve drought resistance, or to understand better the drought resistance mechanisms behind different chickpea lines, and to improve the drought resistance of these plants. Because of course this is another issue with climate change. There is more stresses and different stresses now appearing in all over the world because the climate is changing. There's more drought stress. There are maybe other pests that come. There are pests that come at an earlier time point to the plant and that stress the plants at an earlier growth season, etc. So there's a lot of breeding to do and a lot of improvement.
When you explain this technology, so is this technology expandable? I mean, in the context of India, when we talk about food sustainability, you need to have it this sequencing done. So is it possible to do on a larger scale, where the companies who are producing the plant seedlings or helping farmers to select this particular strain of a plant which already got all the mechanisms and all the checks and balances, which will bring assurance to them that this plant is drought resistant or pest resistant or fungus resistant. So is the technology expandable?
Yes, the technology has expanded a lot, especially because of these technologies that are now more and more widely available, also thanks to companies like ours. We are using a specific technology, but there are also other technologies around using similar approaches, and they are also applied in India on a large scale, of course. So yes, this is becoming more important, and the availability of the technologies is also increasing more important.
Another important aspect which I will like to touch base upon is the costing. So for developing countries, how cost efficient this technology is at this point of time?
I think this goes along with the availability of the technology and the availability of the know-how how to use the technology. And of course, in the beginning, it was still a tremendous cost, but given the higher availability of the technology and the, let's say, more trained people who are familiar with how to use it, that the costs have fallen dramatically. So it is still, of course, costly, but it is much less costly than traditional breeding, because the testing of the traits in different conditions is taking up to 15 years. So it is a very, very long time. And this can be reduced at least, let's say, well, depending, of course, on the trait, down to maybe 3 to 8 years. Yeah.
Can you give us example along with the chickpea where GenXPro have already implemented the technology successfully on a commercial scale, either in Europe or any other country?
Yeah, this the let's say most commercially used application was in rapeseed, where we found a resistance gene towards a specific pest, and this is now currently used every year. The breeders use this marker that we found in thousands or 10,000s of seedlings to understand if they carry this resistant gene or not, and he then takes out the ones that do not carry the resistant gene. So that's one example. Then we have another example for in barley for a virus resistance, yellow dwarf virus resistance, where we also pinpointed or found the genes, markers for the genes. And another example is a more quantitative trait, that is the plant height. So in order to have a high yield or a more practical harvest, the plant height should not be too much, because then too much energy is put into the height growth. So also therefore we are involved in breeding marker identification.
Dr. Rotter, could you please tell us the technology and the brain behind GenXPro, the vision and the science?
Mm-hmm. So we started in 2005 with the vision to look not only at the genomic level, not only at the DNA level, but also to include the analysis of the gene activity. What part of the genome is currently active, what part of the genome is used in a tissue, in a plant, in a biopsy, for example. And therefore, we developed a technology that allowed us to measure the gene expression at low costs at high throughput. And we are since then using the gene expression profiling in addition to classic analysis of the DNA, for example, to understand rearrangements of the genome, to look at mutations. And we take both levels of information and look at both and analyze both in order to better understand the biology or also the, let's say, different biological pathways that are activated in a cell or in a tissue. And we use it, for example, in the case of plant breeding to understand what are response mechanisms of a plant towards a certain stress, or in the field of tumor analysis, we look at what are the pathways that are deregulated in the tumor. And then the second step is that we also try to understand what are possible intervention possibilities, because when we know what pathways are deregulated, what drugs are there to maybe counteract against this deregulation or to kill the cells in the end, because we know the biology much, much better than by simply looking at the DNA.
That's really a leap forward, I must say. Please accept our congratulations on developing the corona detection kit. We learned that GenXPro can detect all the 3 variants in one particular kit in one go.
Yes.
Could you explain us?
Yes, maybe I can start with how this corona pandemic affected us, because also we had first to suffer from the pandemic, because our traditional customers who are breeding institutes or who are medical institutes did not send us any samples anymore because there was a lockdown. And so we thought, as experts in the field of RNA, what else shall we do than to focus on the corona research. So we teamed up with the group here at the university and tried to understand what is the gene expression after cells were infected with the virus. Most of this we were not so much involved in the interpretation, but we helped on the technical level. And then we also started to generate testing kits for the detection of corona. And of course, there were many, many kits around, so we focused on the detection of the variants quite early on, and we were amongst the first who have a very nicely and robust kit to detect the alpha, the beta, the gamma, and the delta variant in a single assay. So this helps to better understand where are which variants now growing. At the moment, only the delta variant is really of belonging and of importance. Sorry, only the delta variant is really of importance, except for a few countries like Colombia, where the mu variant is still very active. Mm-hmm. So we also have a test for the mu variant, and so this was a real breakthrough for us, because we also got recognized by many laboratories who are using our kits now for the detection of these different variants in a single PCR assay.
When you say your kit has a global reach, what were the logistical problems or what were the technical problems you faced in the duration of pandemic while delivering your kits or developing your kits?
Yes, there were big problems to deliver the kits. We had a lot of requests from other countries, from Myanmar to Africa, Northern Africa, South America. And we had, in the beginning, unfortunately, the problem because we had to develop the kit so fast that we could not make a kit that is very stable during a long transport. And we have to ship it cooled. And of course, during the pandemics, there were not enough flights, etc. So that was a big problem. But now, just recently, we now made a new version of the kit so that we can also ship it on ambient temperatures. And so this problem is also fortunately solved. And also there are more flights now, so things are much easier.
So Dr. Rotter, in the field of precision medicine, we heard now the small RNAs are becoming more and more important for the diagnostic purposes. Is GenXPro moving in that direction, and what will be the future vision in terms of technology development?
Yeah, there are two major fields now that are beyond the DNA level. First of all, as I mentioned, is the gene expression, to take this more into account. But there are other layers of information that are very important for good understanding. I'm not speaking now about the metabolomics or the proteomics, because we are experts for nucleic acids. But if we look at this level, there is, first of all, the epigenetic level that is extremely important, because besides the DNA base sequence, there are modifications that direct the gene activity, and one of these modifications is the methylation. So we also have very let's say powerful techniques to understand the epigenetics. The other field is the small RNA, so-called small RNAs or microRNAs, which is one class of small RNAs. And these microRNAs also direct the gene expression and direct the activity of genes and which proteins are finally generated in a cell. So these two I would like to stress as being very important fields where also new diagnostics will be possible. And the most interesting maybe aspect of this is that by looking at small RNAs and also by looking at epigenetic modifications, it is also possible to use so-called liquid biopsies, meaning blood or urine, where nucleic acids are shed into and can be then identified and analyzed in a very high throughput. And we will then generate, or what we're doing is we're generating patterns that are specific for a specific disease or specific for a specific state of a disease, etc. And then can connect these patterns to maybe a certain treatment or maybe a certain, also let's say, no treatment, because we know that a person with this pattern will not respond to the treatment that is maybe usually applied. So we can prevent the side effects because it is known that this person will not benefit from the treatment. So these are the fields that we are also working in, and this whole field can be called or is also called biomarker detection.
One more important thing which I was reading about GenXPro is your ability to detect very small amount of nucleic acid.
Yes.
Maybe if you just
yes.
maybe elaborate a little bit on that topic.
Yeah, this is one important aspect is that usually the amounts of nucleic acids that are available in the bloodstream, or also from small biopsies taken after a cancer surgery, there are very, very tiny amounts sometimes available. So it is very important to be able to understand or to work with these tiny amounts of nucleic acids, both from the liquid biopsies, such as from urine, where very, very low amounts of small RNAs or DNA is captured, or in the case of FFPE tissues or tissue from surgeries, from usually cancer surgeries. And so it's a very important thing. And we have developed also what we invented, and also have a patent for a technology to get rid of or to improve the data after it has been amplified a lot. So that is always required. If you have low amounts, you have to make many, many copies of these nucleic acids. And by doing this, the data can be shifted. There can be a bias towards certain molecules, and we can get rid of this bias using a method that we invented in 2008. It's known under the name of unique molecular identifiers.
So, Dr. Rotter, tell me one thing. In this digital era, most of the entrepreneurs are turning towards making digital platforms, which are computer-based, where not that much of tools, equipments are needed. But when we come to biology, you need a lot of equipments. We just saw the lab setup. It's a huge investment. And how did it motivated you to go to such a high risk
Yes.
maybe business, let's say. So what was the thinking process?
Yes, to be honest, if I look back, there was, of course, it was an enormous risk. But of course, you don't realize that at the time, maybe when you're taking this decision. Still, I think I would have taken it, yeah. And it has to do a lot with personality. If you are feeling, if you have this entrepreneurship ambition or feeling, or not, and I've always had this, and so I was very happy when we were. When I was asked if I wanted to become part of this company that we wanted to found, and so that I took this occasion when I came back from my PhD in Paris, and we started it. And of course, I must say that we in Germany there are possibilities to get a funding for startups, and at the time there was a funding that we received from the state of Hessen and from an institution, a financial institution in Germany that is specializing on financing such entrepreneurships. And so this is what we started with, and where we had the possibility to try our company.
In India nowadays, there are many young scientists which also are looking forward to set up their own businesses and to become entrepreneurs in certain ways. So what could be maybe your word of guidance or thought to become successful?
So one of the most important things is, besides having a good technology and maybe ideally even a patented technology, it is important to inspire the others, inspire the investors, and make them feel comfortable, and that you are trustworthy and you are able to fulfill this. And we see there are many. There are, especially, maybe this is true for the US, not so much for Germany, but there are many stories where a lot of money is put into ideas that are at a very early stage. And so this is what I see a lot, is that there is a lot of money put into ideas if they are sold right and and are able to inspire the investors.
Another thing is maybe how do you say the Indian precision medicine companies as well as precision agriculture companies to come forward and maybe share their problems with you, or maybe to get coordinated or cooperated with the German technology, because the German technology is still considered the best around the world, and we are actually recording this particular video in the context of 70th anniversary of diplomatic ties between India and Germany. So what will be your message to all those companies who are looking forward for getting this technology and bringing good for the country?
If we look at maybe in general, to use such a technology, of course, we would be very happy to use it on every different field of science in collaboration with Indian institutes or Indian companies to find biomarkers, etc. And I think this, if I speak about biomarkers, this is something where I see also a lot of potential for India with a lot of patience. Because one of the most important thing about bringing a product into the clinical use is, of course, the clinical trials. And if there is a good setting for clinical trials in India, I think this could help many good ideas to become finally a product. Because the product, the idea is nothing if it doesn't end up as a product, basically. And the way to this product is in the field of biotechnology and in the field of diagnostics, of course, via the clinical trials. And I think there is a big potential that can be lifted in collaboration with Indian hospitals, both for Indian technologies, but also for German technologies, and collaborations between India and Germany.
We really thank you, Dr. Rotter, for your time and answering all our questions in the most honest and enthusiastic way. We look forward to collaborate with GenXPro and Frankfurt Innovations Center for Biotechnology in future. So thanks a
Thank you
lot.
very much.